Good afternoon, everyone, and welcome to the hold on, to the PRSM MicrobeTalks Exploring the Microbial World webinar series. My name is Angelica Rosado, and I'm the treasurer for the Puerto Rico Society of Microbiology. And this webinar series is focused on introducing professional scientists that particularly exploring their research field and how they contribute to science, not only in the United States, but in Puerto Rico. We have the pleasure to have with us today a ASN distinguished lecturer, Dr. Amariliz Rivera-Medina, and with her presentation titled The Unexpected Protective Features of Novel Antifungal Vaccine Candidate. A brief bio of the Dr. Rivera-Medina is that she is an associate professor at Rutgers, New Jersey Medical School in Newark, New Jersey, and a 2023-2025 distinguished lecturer for the American Society of Microbiology. Dr. Rivera received her bachelor's degree from the University of Puerto Rico, Mayagüez, and her PhD from Rutgers Robert Wood Johnson Medical School. She did her postdoctoral training at the Memorial Sloan Kettering Cancer Center under the mentorship of Eric Hammer, where she began the abiding theme of her research, achieving a better understanding on how the immune system fights fungal infections. After her training, she moved to Rutgers, where she served as an assistant professor in the Department of Pediatrics and a member of the Center of Immunity and Inflammation from 2010 to 2020. Dr. Rivera was an awarded tenure and promoted to an associate professor in 2020. She has also been serving as an associate director for ICC since 2020. Her work through the years has delineated fungus-specific CD4 T-cell responses and monocyte-derived dendritic cells and neutrophil-mediated innate antifungal immune responses in the context of pulmonary fungal disease. Her work has uncovered an unexpected role for interferon lambdas as critical instructors of antifungal neutrophils responses via monocyte-derived type A interferon production in the infected lung. Her career has benefited significantly from NIH efforts to support diversity training, and she has been the recipient of multiple awards under the NCI CRCHD continuing umbrella of research experiences, or CURE. Her research is currently supported by the NIA, which is the National Institute of Allergy and Infectious Diseases, and the Burroughs Wellcome Fund Investigators in the Pathogenesis of Infectious Disease Award. So now with us, Dr. Amariliz Rivera-Medina. Thank you.
Thank you so much. Thank you, Angelica, and thank you, Natalie, and thank you all of you first for the invitation and to all of you for calling in this afternoon to listen to this lecture. As Angelica already mentioned, I study host responses to fungal organisms, and we'll just try to explain to you why we're interested in that. But before that, I tend to pick up speed as I give my lectures, and then I don't want to run out of time before I acknowledge and give thanks to the people that have done the work. So I start with Kei Wang, who is the PhD student, and a lot of what I'm going to show you today was her doctoral work in my lab. And I also want to acknowledge Angelica as a new student in the lab. She's also from Puerto Rico. And Vanessa is a longtime research associate. Ronnie and Laura are postdocs in the lab, and Maria is a new student. I also want to recognize Jorge, who was a graduate student that initiated this project, and some of what I'm going to show you is the data that he generated for his thesis work. And all of the studies I'm going to share with you today are in collaboration with Dr. Chaoyang Xu. And again, just thanking all our funding agencies. All right, so I'm interested in studying host responses to fungal organisms, and I've have the privilege to work in this field now for quite a long time. And when I first started in this field, I have never thought about fungi. And in reality, if you think about it as microbiologists, right, fungal infections are often underappreciated and definitely understudies causes of human illness. So we all know about viruses and about bacteria, and they were appreciated in the general community, but the fact that fungal organisms cause significant mortality and morbidity to various patient populations throughout the world has been underappreciated. In aggregate, fungal organisms actually cause over 1 billion infections worldwide. The majority of those infections are not life-threatening, but there are subsets of patients, especially those that are immunocompromised, that become susceptible to invasive fungal diseases caused by a few pathogens. And together, those fungal organisms cause a burden of disease that's above 1 million deaths per year, which is actually comparable to the burden of these deaths caused by TB and malaria. So the fact that fungal infections and fungal pathogens have been underappreciated has been shifting over the last couple of years. And I'm sharing here with you in this slide a recent publication from the World Health Organization, where they took now finally for the first time the time to dedicate a committee of scientists to study fungal diseases and the impact of human health, and publish a set of recommendations of what the scientific community should be focusing on. And they divided fungal organisms into a critical group, a high group, and medium group of important pathogens. And I'll highlight your attention to the critical group of organisms. And in this list, you'll see 4 human fungal pathogens: Candida albicans, Aspergillus fumigatus, Candida auris, and Cryptococcus neoformans. And the organisms that we've been studying in my lab are Aspergillus fumigatus and Cryptococcus neoformans. And today, my lecture, I'm going to be sharing, starting with our work on Cryptococcus and then transitioning to show you some of our studies in the model of Aspergillus. So what is Cryptococcus neoformans, and some highlights of what the features of this disease. Cryptococcus neoformans is a fungal pathogen that affects humans. It's a yeast, primarily grows as a yeast form. It has a very unique feature that it has a very thick polysaccharide capsule, which has one of the key functions of inhibiting phagocytosis by immune cells. It's found in the environment. It prefers to grow in bird poop and also in eucalyptus trees. That yeast is thought that people acquire spores that the yeast can make, or desiccated cells that are, again, inhaled by the patient. The infection starts first as a pulmonary infection. And one of the key features of cryptococcosis is that it is able to spread from the lung into the blood, and from there go into the brain and cause very lethal cryptococcal meningitis. And in fact, the cause of mortality in susceptible patients is primarily due to the development of these meningitis. And as you saw in my previous slide, it is a high priority pathogen group. One of the key susceptibilities that lead to patients to become, again, susceptible to cryptococcus is dysfunctions of CD4 T cells, primarily HIV-infected individuals, those that do not have access to modern treatments. And when the CD4 T cells come low, then they become susceptible to cryptococcosis and can succumb from cryptococcus meningitis. And there's still a high number of people susceptible, primarily in Sub-Saharan Africa. Options for this deadly disease are limited. Drugs are expensive and often ineffective. And there is a need for better development of drugs. And also, we would like to posit the idea that perhaps developing an antifungal vaccine would be a good option for these patients. And I want to share with you today some of the work that we have done towards the goal of developing an antifungal vaccine. And just because my focus and the focus of my lab is very heavily on the host side of the interaction between fungal pathogens and the host, I wanted just to give you some background. I'm going to be using a lot of immunological terminology, so hopefully this will help all of us to, and I'm not sure of what all of your backgrounds are. So I just wanted to try to, bring us into context of what some of the terms that I'm going to be talking about and what is known in the field before. So Cryptococcus, as I mentioned, affects patients with immunosuppression, especially those that lack CD4 T cells. What many studies, primarily in mouse models of disease, have demonstrated, but also from clinical observations in humans, lead us to have the following model. That when Cryptococcus neoformans gains access into the host, there are innate cells, which are those that are early responders of infection, that come and recognize the presence of the fungal infection, and that there are key roles by dendritic cells and alveolar macrophages, and those cells pick up antigen from the Cryptococcus neoforman E cell. They present those antigens to T cells. T cells come out of the thymus as a naive inexperienced cell that has the option of differentiating into various subsets of T cells. And these include, very importantly for this infection, Th1 type of T cells that are characterized by the production of interferon gamma, which is a key effector cytokine. And you're going to hear the word interferon gamma a lot. So this is a cytokine, again, that is made by Th1 cells, also by CD8 T cells. And those cells then act on macrophages and other innate cells to mediate eradication of the fungus. And what studies in mouse have very nicely demonstrated is that if you infect a mouse and the mouse develops a strong Th1 response, as well as with support by Th17 cells as well, then the infection can be controlled and mice can be protected from infection. On the contrary, if the host immune response is tilted towards a Th1 dominated immune response in which naive T cells differentiate towards Th2 cells that are characterized by the production of different cytokines, in this case, cytokines IL-4, IL-5, and IL-13, then that's a detrimental response. It has high eosinophilia. The Cryptococcus cells continue to proliferate in the lung, and they disseminate from the lung into the brain tissue to cause deadly meningitis. And again, this is very nice correlations between clinical observations in people and something that can be modeled in mice. All right. So when I started this project, it came from a collaboration with my collaborator, Chaoyang Xu, who I mentioned before. I will call him Chao from here on. It's a lot easier to say. So he is a fungal pathogenicist. That's his expertise. That's what he studies. And he's been studying Cryptococcus for a long time. And one of the things that they do in his lab is that they make mutants of Cryptococcus to try to understand what are the pathways and what are the genes that influence the virulence of Cryptococcus. And in previous work, he had identified this gene that is called FBOX1 protein, or FBP1. And you're going to hear that term a lot. So he found this gene. He made a knockout mutant, deficient strain of Cryptococcus neoformans that lack this FBP1 gene. And you're going to hear again that he'd kill FBP1 mutant. And this is the primary data that he published before we started our collaboration. So what he found is that if he knocked out this FBP1 gene, which is shown here, an infected mouse, and the mouse survival is shown here in the solid bar, those mice were protected from infection. And that is in contrast to the parental strain of Cryptococcus H99. And this strain is very virulent in mice. So mice without any immunosuppression, if you infect them with this H99 clinical strain, and mice rapidly succumb to infection. And previous studies have nicely demonstrated that there are three main critical virulence factors in Cryptococcus. These include the formation of capsule, which I mentioned in my introduction, is this thick polysaccharide capsule that prevents immune recognition and phagocytosis. And what Chao previously saw is that his mutant FBP1 is a virulent in vivo, meaning that it does not kill the mice, but it still has capsule. So the reason why this mutant was a virulent is not because it lacked capsule. He looked also at melanin, which again is another important virulence factor. The mutant was not affected in that virulence factor. And the third major virulence factor is the ability of Cryptococcus to grow very well at 37 degrees. And as you can see here, the mutant also grew very well at this temperature. So when I joined Rutgers, he was already working and had published this, and asked for a collaboration to see if we could figure out whether a virulent phenotype of the FBP1 deficient strain was linked to any changes in the host response. So we started then investigating what this mutant triggers, and could it be that there's some enhanced host immune responses to this mutant that allows the host to survive. And so what is this FBP1 that I'm going to be talking a lot about? This protein is an FBOX1 protein. It's part of this ubiquitin proteasome system. It is a component, is a E3 ubiquitin ligase, meaning that is this unit of the component that recognizes different targets for ubiquitination and further proteolytic degradation. And so this mutant, and just so that we're all in the same context, this mutant actually has targets because it's one of the FBP1, is an FBOX1 knockout. It actually results in changes in protein levels of multiple targets. And so one of the big questions for the future for our team is to try to understand which one of all the multiple targets that FBP1 has is responsible for the immunological phenotypes that I'm going to share with you. And that's the work that they do in Dr. Chow's lab. What I'm going to show you is more of all the immunological characterizations that we have done to the mutant and how we are using this mutant as an interesting and what we consider very novel and promising antifungal vaccine. All right, so to summarize multiple publications that we had before, what we uncovered in our initial studies with this mutant was the following. So I already earlier mentioned to you, I introduced you that what is thought about important immune responses to Cryptococcus. What has been found previously in the field is that when you have a very virulent Cryptococcus strain, such as H99, what happens in the host is this aberrant and a non-productive immune response in which is characterized by high Th2 cells and very rapid dissemination of the yeast from the lung into the brain. What we uncover is that the mutant, the FBP1 mutant, had an altered immunological profile, and now it was characterized by high recruitment of CCR2 positive monocytes, neutrophils, monocyte-derived dendritic cells. These early innate responders helped skew the response of T cells toward a Th1 type phenotype, including also strong Th17 differentiation, high production of interferon gamma, and prevention of dissemination from the lung into the brain. So these immunological features were very intriguing, again, that it was changing the parental strain into now a protective type of host immune response. So with this knowledge at hand, we decided to test whether we could exploit this increased immunogenicity of the FBP1 mutant as a way to develop a new antifungal vaccine. And what we decided to do was the following, was to take the FBP1 mutant and prepare heat-killed versions of this whole E cell, and then deliver this as a pulmonary vaccination. And the idea of why we decided to use heat-kill preparations is that, as I mentioned in my introduction, the susceptible patients that succumb to cryptococcosis are those with immune defects. And we wanted to be able to have a vaccine that doesn't have any in vivo replication of the strain, even if it's an virulent strain. In the context of immune suppression, you could envision that that strain could cause disease in a patient if they don't have a normal immune system. While a heat-kill preparation is non-replicative, it's not going to proliferate and is not expected to cause any illness. So we took the FBP1 mutant, heat inactivated it, and then confirmed that, of course, in vitro before administration to the mice. And all the vaccinations are done in a pulmonary delivery. And so this is the time frame and the experimental setup of all the data I'm going to be showing to you today. We immunize early on. We let the mice rest. Then we give them a boost vaccine. And then 14 days after the second boost, we then challenge the animals with live virulent H99 and to look at that way at protection. Throughout my talk, I might end up referring to early induction phase as compared to later effector phase, meaning, and what I mean by that is that in the early induction phase is when we're first immunizing the immune system. And then at the later effector phase is when we challenge with the live infection and looked at whether these initial changes in the host response have any effect on protection later on. All right. And again, in the interest of time, I'm going to show you a summary of what we have uncovered and how we determine that indeed this is a very interesting and potent antifungal vaccine. So with that, a schedule of vaccination that I mentioned to you before, what we found was the following. Indeed, this vaccine can be a very potent protection against homologous defense. And when I use the term homologous, what I mean is the following. As I showed you, this FBP1 mutant is based on the H99 strain and is a Cryptococcus neoformans vaccine base. And when we challenged with Cryptococcus neoformans parental H99, we saw 100% protection in the animals. And that is in contrast to the susceptibility seen in mice that have not previously vaccinated, which rapidly and reproducibly succumb to Cryptococcus infection in vivo. So answer number one, yes, this is a potent vaccine that can induce homologous protection. Beyond this, we wanted to ask the question of whether we could use this vaccine for as protection against other fungal organisms. And this is where the unexpected part that I refer to in my title is alluding to. And that is that we tested whether this Cryptococcus neoformans based vaccine could protect against a sibling species of Cryptococcus, and this is Cryptococcus garii, is in the moderate risk group of pathogens in my earlier introduction from the WHO. And what we observe is, intriguingly and very interestingly, that vaccination with this neoformans-based FBP1 mutant led to protection against challenge from C. garii. As you'll see, unvaccinated mice here on the control, black line, all of them succumb to C. garii infection. And this is very interesting and unique. And just to give you context, because of the following. As you all know, and you probably all were forced to take the COVID vaccine, and you take the influenza vaccine. And when you're kids, you're giving, measles and mumps vaccines. Each of those vaccines will only protect you against the pathogen that they were made for. For example, in the COVID vaccine, only protects you against COVID, and most often it only protects you against very specific subtypes of that virus. A COVID vaccine cannot protect you against a bacteria, and it cannot protect you against even other viruses, right? And that's the way that all approved vaccines work. This is a novel finding in that now we have a preparation of one pathogen, which can protect against a parental pathogen, which is a more common, but beyond that, it can also protect against a different kind of fungal organism, and in this case, a sibling, meaning highly related organism, but a distinct species. Moreover, and what I'm going to get at the end of my talk to show you is that we also discovered that this Cryptococcus-based vaccine can actually be a potent vaccine against aspergillosis, which is a totally different fungal organism. This is a mold organism, and we're going to get to that closer towards the end of the talk. So we discovered that this can also confer what I'm going to refer to as heterologous protection, meaning a protection against a different organism from the original one that the vaccine was based on. So all of these studies are done, again, as a typical vaccine regimen. We immunized, we boost, and then we challenge with the pathogens, and the vaccine works very nicely. In addition to that, we have also discovered that the vaccine can be used therapeutically. And in this setting, what we do is we take the mice, we infect them with H99, we let the infection go on for 3 days, and after that, we can then intervene and give the animals the vaccine. And the mice are also protected in that setting, meaning that it can also have a potential as a therapeutic. And we're all very excited about this. Okay, so and just to summarize some of the mechanistic work that we had done before, I want to take the time to show you primary data for a paper that we just published yesterday. And so I'm just going to summarize what we discovered before. So I showed you we have a potent vaccine. It induces homologous and heterologous protection. And then we are devoted to understanding how does this vaccine mediate this protection. And what I'll summarize for you before some key points for what we're going to be discussing now is the following, is that the vaccine requires interferon gamma as an essential mediator of vaccine-induced homologous protection, meaning crypto to crypto protection requires this important cytokine. And the way we do that is that we took interferon gamma receptor deficient mice, vaccinated them, and uncovered that those mice could not be protected. Now, I mentioned in my introduction that susceptible people, especially humans, are those that have dysfunctions on CD4 T cells. And how in the world am I going to tell you that we're making an antifungal vaccine if people without CD4 T cells cannot be protected? We needed to know, is this vaccine able to confer protection in a CD4 deficient host? And we demonstrated indeed that that can be the case. what we found before is that if we deplete CD4 T cells, the mouse can be protected by this vaccine, and the protection depends on CD8 T cells that expand and make interferon gamma in the host in the absence of CD4 T cells. We also showed that we could deplete CD8 T cells, and CD4 T cells alone were sufficient to confer protection. But if we take a mouse and deplete both CD4 and CD8 T cells, if both of those T cell populations are gone, then the mouse is no longer protected. So this tells us that lymphocytes are required, specifically T cells. It can be either CD4 or CD8s, and it requires interferon gamma. Now, from those published observations, our next question, and what I want to take some more time to discuss with you, is we wanted to understand, okay, so now we know interferon gamma is required, but we want to know exactly what are the mechanisms of interferon gamma mediated protection. We wanted to identify and validate what are the important sources of interferon gamma, especially those early sources after initial vaccination. And we also wanted to know what are the important targets of interferon gamma at the later phases of protection. And again, just to orient you on some of the terminology that I'm going to be using, we're going to be investigating then what are the roles of interferon gamma early in this, what we call early induction phase after initial vaccination. And I'm going to show you data after mice are immunized against in the lung at day 3 and day 7, and also looking at later on at 4 and day 7 after the challenge with H99. Okay, so that's what we call the late effector phase, when the mice are actually challenged with the virulent pathogen. All right, so one of our questions was, what exactly is interferon gamma doing early on after immunization? First of all, is there any role for early interferon gamma after vaccination? And if so, what exactly is happening? To address this question, what we did is employ wild-type animals and then induce inactivation of interferon gamma by using antibodies that neutralize the effect of interferon gamma, meaning that the mouse is able to make the cytokine, but the antibodies bind to the cytokine and prevent it from binding to its receptor and having the effector function on the target cells. And what we did is do a sustained interferon gamma neutralization with XMG 1.2 antibodies to again neutralize the effect of the cytokine, and looked at day 3 and day 7 at our metrics of host responses. I mentioned to you before that in our earlier studies, we had uncovered that monocyte-derived dendritic cells are very important for the orchestration of this protective response to the FBP1 mutant. And what we observe is that when we neutralize interferon gamma earlier on, we saw that the development of MO disease was significantly impaired. In gray, in black here are control mice, and the purple ones are those that are interferon gamma neutralized. And what we saw is that was actually a significant reduction in the appearance of monocyte-derived dendritic cells in the lung. Importantly, I also mentioned to you that we know that our vaccine requires T cells, and specifically gamma-producing T cells. So we assayed those responses and observed that when we looked at Cryptococcus-specific T cells in the draining lymph nodes of the vaccinated mice, we observed that if we neutralize interferon gamma within this first week of time frame, what we saw is that was actually reduced production of IL-2, and that is a surrogate for activation of T cells, active proliferation of the cells. Importantly, the T cells were reduced in their ability to produce interferon gamma that I mentioned to you, we know is a required cytokine. And in contrast, what happened is this skewing away from this protective Th1 response towards a Th2 response that we measured as IL-5 producing T cells. So interferon gamma is important early on to induce and educate T cells that are going to be protective later on, and are they themselves going to become important sources of interferon gamma later on. Since this is an early time frame of a response after vaccination, we wanted to know what are the early sources of interferon gamma prior to that education of T cells that are then going to become the later sources of interferon gamma. And we started looking for, again, what is first? The first question was, what is the kinetics of interferon gamma production early on within the first 7 days? And what my student KE discovered is that there's this kinetics of induction of interferon gamma, where there is indeed innate sources of interferon gamma. And you can see that the initial peak of interferon gamma production, both at the RNA level as well as the protein level, is at day 3 after vaccination. She employed intracellular cytokine staining assays to determine what were those innate cells that were making interferon gamma, and identify some intriguing sources of interferon gamma in the form of monocytes, which are early responders, as well as neutrophils. And that was interesting to us because these innate cells are usually not considered important sources of interferon gamma. It is thought that in other infections, interferon gamma is primarily derived from natural killer cells. And we are seeing here that in our model after vaccine, we see monocytes and neutrophils as being able to produce interferon gamma. From that observation, we wanted to test, okay, we see the cells making interferon gamma, but are they important sources of the cytokine? And in order to ask that, what we did is test what would be the impact of removing each of the cell populations to our vaccination response, and specifically looking if we remove neutrophils, what happens to the activation of protective T cells. And in that case, what we did is use antibody-mediated depletion of the neutrophils using an anti-Ly6G antibody. And we confirmed that there was effective depletion of the neutrophils, and then looked at what happened to the levels of interferon gamma in those neutrophil-depleted mice, and observed that indeed removal of neutrophils resulted in significant reductions in the global transcription of interferon gamma, as well as significant reductions in the total protein levels of interferon gamma in the lung tissue after vaccination. And this data demonstrates that indeed neutrophils can make interferon gamma and are an important source of gamma, because when we deplete them, the levels of interferon gamma are significantly reduced. In turn, consistent with the data I showed you in the previous slide, when we remove neutrophils and interferon gamma is low, now we also see those reductions in MODCs, which is consistent with what we saw with the interferon gamma neutralization. So together, this data suggests that early interferon gamma is important for MODCs, important for Th1 differentiation. In turn, neutrophils themselves are important sources of gamma, and without them, there's reductions in the gamma levels, as well as reduced differentiation of monocyte-derived dendritic cells. To further investigate whether monocytes themselves were also important sources of interferon gamma, we turned to a model that we have used in the past for our study of antifungal immunity, and it's a genetic model in which we are able to induce the selective depletion of CCR2 positive monocytes. This is a mouse that has a genetic modification where we have placed a diphtheria toxin receptor under the control of the CCR2 promoter. Mice are naturally resistant to diphtheria toxin, and cells that express the diphtheria toxin receptor become susceptible. In this case, our CCR2 positive cells express the receptor. And then what this allows us is to be able to specifically induce the depletion of those cells depending on the time and duration in which we administer the diphtheria toxin. So what we did was deplete monocytes before immunization and after immunization, and ask what happened at this day 3, which I showed you is the peak of interferon gamma expression after vaccination. And what we observe is that similar to what we saw with the neutrophil depletion, if we remove monocytes, they also had lower levels of interferon gamma RNA, lower levels of interferon gamma protein. In terms of the T cell response, we could see that there was reduced recruitment of both CD4s and CD8s when we depleted monocytes. In terms of CD4 T cell differentiation, we saw that they have reductions in interferon gamma production, and accompanied with increases in IL-5. Again, a consistent switch from a protective Th1 response towards a detrimental type 2 immunity. In terms of recruitment of T cells to the airways of the vaccinated mice, we also saw that there was significant reduction in the presence of those T cells. So altogether, these studies with the CCR2 depleted tells us that monocytes are also important contributors to the global amounts of early innate interferon gamma, and in turn, that is required for the differentiation. Further, also based on previous work that we had done, in which we showed that monocyte-derived dendritic cells are important for antigen transport to the lymph node and priming of T cells, we also hypothesized that the contributions of monocytes in here is linked to that monocyte-derived dendritic cell population, which is required for the expansion of T cells and recruitment of those cells to the airway, where they're going to then be poised to protect the mouse from challenge from H99. All right, so I've been talking to you a lot about monocytes. What are these cells and what else can they be doing? Monocytes are precursors, myeloid cells that reside in the bone marrow primarily at the steady state. As I mentioned to you, they express the CCR2 receptor, and they are highly recruited to the periphery and to the infected tissues in the context of vaccination, in the context of infection. They are key players to protection against multiple kinds of pathogens, bacteria, viruses, as well as fungi. And importantly, monocytes are precursors of tissue macrophages and can give rise to dendritic cell subsets, which I have been alluding to as what we call monocyte-derived dendritic cells. So in the data that I showed you in the earlier slide, when we deplete monocytes in the CCR2 depleter, we are depleting the precursor, but we also depleting all the derivative cells that would have differentiated from those CCR2 positive cells. I also mentioned to you that our vaccination scheme starts with an immunization at day -42, resting the mice for 30 days, and then boosting the animals for 14 days. And one of our questions for this study was, what is happening to monocytes after vaccination? Can we identify monocyte-derived cells? And are those cells important targets of the effects of interferon gamma? And to ask those questions, one of the challenges in the model and the system is that once a monocyte precursor differentiates into these macrophages and dendritic cells, they lose expression of the CCR2 receptor, and it becomes very difficult by traditional flow cytometry to be able to identify what are the monocyte-derived cells. But scientists have developed this cool system, which is called a fate mapper model, in which you can genetically label monocytes prior to any immunization. And then you can track the fate of the monocyte-derived cells by using this fate mapper, which is works in the following way. So you have a tamoxifen inducible Cre protein, a Cre enzyme, and the expression of this Cre enzyme is limited by whatever promoter you place that gene under. In our case, we have access to two different fate mapper models, one that is driven by the CCR2 promoter, as I mentioned to you, which is expressed by the monocytes, and also a CX3CR1 commercially available inducible Cre, which is also a chemokine receptor expressed by monocytes. So you have your Cre line, right, that is going to express the enzyme. Then you breed that animal to a target mouse that in this case has a fluorescent protein that is controlled. Ahead of it has a stop codon so that this fluorescent tag cannot be expressed in a mouse without a Cre enzyme. When you breed these animals, and because this is inducible, once you inject the tamoxifen molecule to activate this Cre enzyme, then it can go into the locus, remove the stop codon, and turn on the green protein. In our case, we use an R26 TD tomato, and our offspring mice are red once we give them tamoxifen. And in this case, we are labeling the monocytes prior to vaccination, and then turning them red, and then following what happens to the monocytes after vaccine. Do they become macrophages? Do they become dendritic cells? And we can also look at what are their responses after challenge. I will also take the time in this slide to explain to you that some of the models that we have also used after this kind of experiment is using mice that are Cre flocks for targeting genes. And in that case, you have a mouse that has a constitutive Cre under a promoter of interest and a target gene that has flank flux sequences. And in that case, you can use the animals to generate a conditional knockout in which you remove gene expression from your target immune cell of interest. All right, so we use our fate mapper mice, and then we vaccinated them. We let them be there for 30 days, and then we ask, can we find monocyte-derived cells? And just to orient you, we use a lot of flow cytometry to identify the cells that I've been talking to you about. This is a representative FACS plot for lung tissue, where we use these markers Siglec-F and CD11C to identify alveolar macrophages, which are double positive cells. And also you have the CD11C positive cells here, which are comprised with a mixture of monocyte-derived dendritic cells as well as other dendritic cell populations. And if we use fate mapper mice, vaccinate with HIP-KILL-FBP, and we looked at different times. We looked at day 10 and day 30, and I'm just showing you representative FACS plots where we observe that if we looked at this alveolar macrophage gate 30 days after vaccination, we see a robust 35% of the cells are red, meaning that they came from monocytes. And what has happened is monocytes come into the lung after vaccination, and they give rise to alveolar macrophages that are now of monocyte-derived origin. Cells that remain dark means that those are true tissue-derived alveolar macrophages. We also saw that there's dendritic cells that are red after immunization. And as a control, we see that eosinophils, for example, are not red in either population. They're not expected to be in this model. They would not be labeled. All right, so with these tools, now we know, yes, monocytes are coming in and they're giving rise to alveolar macrophages. So we wanted to understand, are they changing over time, and are there targets of interferon gamma? We see that indeed alveolar macrophages continue to increase after vaccination while the dendritic cell population stays steady. So what we decided to do is focus on this alveolar macrophage population. We sorted the cells. We did a transcriptional analysis. We see that they have distinct profiles and have this separated in this plot. And compared to naive cells, what we observe is that indeed monocyte-derived alveolar macrophages and tissue-derived alveolar macrophages have signatures of interferon. These are all interferon-inducible genes, meaning that this data suggested that these two kinds of CD11C positive populations were actual targets of interferon gamma and retain a sustained transcriptional response for 30 days after vaccination. This gave us the idea that CD11C positive cells, which is comprised again about this mixture of dendritic cells and alveolar macrophages, could be important targets of interferon gamma for protection. And in order to investigate that possibility, we turned to a STAT1 knockout mouse. And what I wanted to use this slide to remind you is that I've been talking to you a lot about interferon gamma, and just very briefly to tell you that this is the receptor complex, and the cytokine requires this transcription factor called STAT1 in order to activate interferon-inducible genes that would have shown up in our transcriptional analysis that I just showed you. So what we did is use a Cre-Flux system in which we are using a CD11C Cre mouse crossed with a STAT1 flux. And like I mentioned in my previous slide, this means that we're doing a conditional removal of the STAT1 transcription factor in CD11C positive cells. We vaccinated and boosted those mice and then challenged them with the H99 and tracked responses over time. What we observe is that if we remove STAT1 from CD11C positive cells, the mice cannot be vaccinated and are as susceptible as unvaccinated animals. And this is in contrast to vaccinated STAT1 flux mice. And the mice that are susceptible also show weight loss. When we look at the fungal burden, we observe that in contrast, or you'll see, as we've shown you before, the vaccinated mice have very strong protection. They have lower CFUs. They prevent primarily dissemination to the brain. There's no fungal colonies in the spleen. Unvaccinated mice, which are shown in white here, have high fungal burdens in the lung, in the brain, and the spleen. And the C11C Cre-Stat mice actually had similar fungal burden as unvaccinated mice, demonstrated that indeed CD11C positive cells require STAT1, meaning that there are important targets of the protective effects of interferon gamma. And just to summarize, again, what we think is happening is that we think that during this FBP vaccination, we get high recruitments of monocytes and neutrophils. Both of those cells are important early sources of interferon gamma. And this cytokine is important for activation and differentiation of monocyte-derived dendritic cells. In turn, those monocyte-derived cells are important for the proper expansion and activation of protective Th1, as well as expansion of CD8 T cells. Both of those cells are then important later on during the late effector phase as sources of interferon gamma. In turn, that interferon gamma needs to act on innate cells, primarily CD11C positive cells, as our CD11C STAT1 experiments demonstrate. And our transcriptional analysis suggests that within those CD11C positive cells, we have important contributions by monocyte-derived alveolar macrophages, as well as by tissue-derived AMs. And that we think that these, again, are important direct eradicators of the Cryptococcus cell. All right, in the last few minutes, I want to share with you, go back to Aspergillus fumigatus and this intriguing observation, again, unexpected observation, that the vaccine that I've been talking to you in the homologous protection can actually induce protection against Aspergillus. Aspergillus fumigatus is another inhaled fungal pathogen. It makes very small fungal spores that get inhaled and cause invasive disease in patients with immune dysfunction, especially patients that have low numbers of neutrophils or defects in NADPH oxidase, which is a mediated reactive oxygen species. And this ROS is an essential effector function of innate cells to control aspergillosis. In our previous work, we found that when we do a vaccine, boost, and immunosuppress mice with cyclophosphamide, those animals can be protected. Let me back up 1 minute and say that mice are highly resistant to Aspergillus in the normal context of a normal immune system. Humans and mice, then that's the case for humans. As I mentioned in my introduction, only immunosuppressed patients become susceptible. Cyclophosphamide is a drug that is also used in people, and it induces very strong neutropenia in mice, and is a way to model susceptibility to aspergillosis. So if you take an unvaccinated mouse and treat it with cyclophosphamide and infect it with aspergillus, they become highly susceptible and they die. In contrast, if we took those mice and pre-vaccinated them with the hit-kill FBP at days 42 and 14, the mice are fully protected from invasive disease, they don't lose significant weight, and we prevent the development of this invasive fungal growth that we can monitor in histology by GMS staining. And now what we've been trying to do, and all of this part of the talk for a few slides, is going to be unpublished observations of our attempts to try to understand how is this vaccine working in this heterologous model of protection. So our question number one is, okay, this works with this R21 strain. Is this broadly applicable to multiple clinical isolates of Aspergillus? So we did immunization, boost, cyclophosphamide immunosuppression together with challenges with 3 different clinical isolates of Aspergillus that have different virulence ranges. And what we observe is that our vaccine can indeed be a potent protector against challenge with Aspergillus fumigatus of 3 different isolates. And again, all vaccinated mice are protected from this weight loss, in contrast to all the mice that succumb to infection if they are unvaccinated. Our other question was, are T cells involved in this protection? I showed you that in the model of homologous protection, T cells are very important. And we wanted to know, in defense against heterologous challenge with Aspergillus, are T cells involved? To address this, we vaccinated, we boosted, we immunosuppressed the mice with cyclophosphamide to make them susceptible to Aspergillus. And on top of that, we're adding a T cell depletion using anti-Thy1.2 antibodies. And what we observed quite strikingly is that immunosuppressed and T-cell depleted mice are protected from invasive disease if they have been previously vaccinated. So this tells us that this heterologous protection induced by our vaccine is mediated in a T-cell independent manner, which is quite surprising, and another one of the unexpected features that I allude to in my introduction about what are very different features of our vaccine as compared to others. Furthermore, we wanted to understand, okay, so if the vaccine is not affecting and doesn't require T cells, it must be, is it affecting innate responses? Is it making innate responses better? And to ask that question, we have access to a great tool that was made by our collaborator, Tobias Ho, in which we have a fluorescent strain of Aspergillus. It's a viability readout strain that is double red and blue when the spores are alive. And once the immune cells phagocytose them and kill them, they lose their red signal, but they remain blue. And using this, we can make calculations of efficiency of killing in vivo in the host. And what we observe is that when we pre-immunize the mice with the hit-kill FBP, what we observe is that the innate response of monocytes, of neutrophils, and alveolar macrophages is indeed boosted by the vaccination with hit-kill FBP. And that helps to control a fungal burden. So our ongoing hypothesis is that this vaccine is able to activate innate responses in a manner that makes them really efficient at being able to control the fungal spores. In the last 2 slides, then we're trying to now understand, okay, which one within all of these innate cells are the ones that are mediating protection? I mentioned in my introduction that neutrophils are really well appreciated as key mediators of protection against Aspergillus. Cyclophosphamide is resulting in a significant depletion of neutrophils, but that depletion is not absolute. So we wanted to know, is the vaccine able to change neutrophils in a way that they can protect, or are they dispensable for our protection? So what we did is immunize, boost, a cyclophosphamide treat, and alternate that cyclophosphamide treatment with neutrophil depletion to remove the cells and ask, are the mice protective or susceptible in our vaccine model in the absence of neutrophils? And quite strikingly and unexpectedly, another unexpected observation is that we can remove neutrophils in this model and the mice are still protected. So whatever the vaccine is doing to innate cells does not require neutrophils. And the last data set I will show you is, I mentioned CD11C positive cells earlier. We know in the homologous protection they are important. So we wanted to know, is the heterologous protection that we are activating against Aspergillus mediated by CD11C positive cells? So what we did is take a diphtheria toxin depletable CD11C model in which we can use diphtheria toxin to kill the CD11C positive cells, immunize, boost, immunosuppress with cyclophosphamide, infect with Aspergillus, and then alternate depletion of CD11C positive cells. And what we observe is that if we remove CD11C positive cells, the protection is gone, and those mice lose weight and succumb to infection. So this has now shown us that CD11C positive cells are the key mediators of this heterologous protection. And we're in the process of trying to understand what are the mechanisms, how are the cells protecting. We have some interesting ideas about this being a form of a memory of innate cells, and a lot remains to be discovered. So my very last seconds here with you guys before we take questions is to share, we're actively looking for people in the lab for postdoctoral positions. So I would love to hear from you if you're interested in our work and would like to come to our campus. This is our medical school in the Newark campus. My lab is here in the Cancer Center. We're very close to New York City. I also share with you that I am the director of an undergraduate summer research program at New Jersey Medical School, and we are actively recruiting undergraduates to come for summer for 10 weeks to do hands-on research at our university. We also have a great multidisciplinary PhD program for those of you that are looking for going to graduate school. I encourage you, get in touch with me if you want more information about our programs. And again, thank you for your attention, and I would love to take your questions.
Thank you very much. Orlando, let me unmute you.
Hi, can y'all hear me?
Yes.
Awesome. Thank you, Dr. Rivera. I was wondering, what drove you like towards this type of research, like vaccine research, and these fungi specifically, like why fungi?
Yeah, so as you will likely find on your own journeys, for my personal experience has been just what opportunities have come my way and saying yes to the opportunities that have come my way. So I went to the United States, to the only university that gave me a chance to do a PhD. And from there, I went to the only lab that gave me a postdoc. And in that postdoc, the PI had a project that nobody wanted, which was to work with these fungi, Aspergillus. And his task to me was, I want to make a T-cell receptor to Aspergillus. And, nobody else wants this project. Do you want it? Like, sure, I'll do it. And it worked out really great. And that's how we ended up, studying fungi. And it has been a great pleasure to do so. I've been part of the growth of this field, and I really liked it. And specifically about vaccines and crypto, also by chance, Dr. Chow, I was working, and I still have a large part of my lab working on aspergillosis and mechanisms of protection. And he came to us to start studying Cryptococcus because of the mutant, and that led to the vaccine. And here we are. And I think it's a very interesting finding. We're very hopeful. We have a patent on the vaccine. His lab is very heavily involved in trying to, again, understand more targets of FBP to make a more purified version of the vaccine rather than having this whole cell preparation. So a little bit of chance. And again, just taking all the opportunities that have come my way to say, I'm just going to follow these opportunities. Thank you for
I
your
love that. Thank you.
If anybody else has a question that they want to ask Dr. Rivera, you can type it in the chat, or by all means, you can unmute yourself and ask.
Yes. I think I see a question here. How did you find that it was CD11C positive cells and not another cell? All right. So a lot of how that happened is by narrowing down of options. And so just for the interest of time, I didn't show you everything that we have. Please look at MBio. Our paper was published yesterday in MBio, an ASM Press journal. And so there's a lot of data there where we rule out other cells. And yeah, basically by ruling out other cells, we ended up with CD11C positive cells. It's the one that gave us the phenotype that they were important. And the reason why that marker is because it's a marker expressed by alveolar macrophages. And if you look at reviews on what are important effectors of Cryptococcus control, alveolar macrophages and macrophages are thought to be key mediators of direct inactivation of the Cryptococcus cells. So it was a good target to go for, to study, to investigate. Thank you.
Thank you. And does anybody else have a question? If not, I have a question of my own. I wanted to ask you, and you kind of mentioned it in your presentation, but I was interested to know how long does the protection of the vaccines that you guys have done, both with the Aspergillus and the Cryptococcus, how long does the protection last? And I saw that you do boosters, so how much days in between?
Yeah, excellent question. So for the crypto to crypto protection, then this has been done primarily in Dr. Chow's lab. They have taken the mice, tried different distance or different time frames between the initial vaccine and the boost, and then after the boost, do different time points after to challenge with H99 at different time points. And they have shown that even after the second boost, they can do a couple of months after, and the mice are still protected. So that's for the crypto to crypto. For the crypto to Aspergillus is something that my lab has been primarily leading. And what we have done is we wanted to know, do we really need a boost? Can be one shot, is one shot sufficient, and how long? So now we know that one vaccine is enough. We know that the vaccine can work quickly. So at 14 days, it works. It works at 30 days. So we can just do one boost, one vaccine, no boost at 30 days, and it protects. And that's one of the ongoing experiments in the lab. So we have mice that are parked for 3 months, 2 months, and we're trying to do the kinetics to see what's going to happen, to see how long it lasts. And this is very important for the Aspergillus protection because it's an innate cell-driven protection. So T cells are known to be very well long-lasting. So when a vaccine depends on T cells, you can really envision that they can last a long time. But when the vaccine depends on innate cells, it's an open question whether they will last a long time. So yeah, we need to find out. We're very interested in that. Thank you. Yeah.
Thank you. Miguel wrote a question. He asked, have you thought of using histoplasma?
It's a good question. We would love to. One of the challenges of testing our vaccine with other pathogens is finding collaborators that can, and are able to do it. This work has to be done in biosafety, level 2 conditions, and our expertise. I have expertise with Aspergillus and Chow expertise with crypto, so that's why we tested those strains. In one of our MBio papers in 2019, he also tested Candida because we had a collaborator that worked with Candida. And in there, we saw some prolonged survival, but all the mice eventually died. So it wasn't as robust as protection with Garii or Aspergillus. But yeah, we would love to test histo. And actually, there's a collaborator that now has access to coccidioides. So we would like to test that, a fungal pathogen as well, just trying to see, how broad can it be?
Thank you. One quick question would it be okay if we share your email to the students in attendance?
Please share it with everybody. Contact me, especially if I said you're interested in the summer program, more information about graduate school. or if you guys want to come by my lab, if you're interested in postdocs, or even for your faculty. I don't know if there's faculty here, but if anybody wants to come and do sabbaticals in Rutgers, I'll be happy to be your point contact for more information.
Thank you so much, Dr. Rivera. And with that, we thank you for your time. We also want to remind the attendees that there's an evaluation form in the chat, if you could fill that out. And on Thursday, we have another speaker, Dr. Plus Kumba. She's going to talk about science policy and how to dive into the world of diplomacy after the PhD. So thank you so much, Dr. Rivera. Have a great afternoon. And to all the attendees, thank you so much for tuning in.
Thank you all. Have a great night.